under host and pathogen interaction. The host over here is Pisum sativa, that is garden pea, and the pathogen over here is a biotrophic pathogen named Iricipe piscis. Biotrophic name itself indicates that the pathogen requires living organisms for its survival. So before dwelling into the topic of my talk, I just want to introduce the host. The host over here is garden pea. So garden pea, which we use in our culinary practices on a regular basis, it is a source of proteins, fibers, minerals, nutrients. So this garden pea plant belongs to Fabaceae family, and it's a small plant which grows to approximately 0.75 meters above the ground, and it's a full season crop. So these inexpensive crops, we, as I said, that we use in our culinary practices, and it is a resource of proteins, fibers, minerals, and it is it fulfills the dietary requirement of a malnourished person because of vitamins, minerals, and all those things. That will fulfill all the requirement of a malnourished person. Even a person like me, overweight person like me, if I wanted to put down my weight, I just have to incorporate garden pea in my regular diet. So apart from that, when we see the protein content of garden pea plant, the protein content of garden pea plant is 5.42. So apart from this thing, astonishing things have been come to my notice when I started reviewing the literature. So garden pea plant has very important role in providing cardiovascular function. It is having an anti-tumor function it provides, and apart from that, it prevents cancer, and it is having very different various properties like anti-diabetic and anti-hyperlipidemic and cytoprotective properties. So what actually the problem is, we are having a garden pea plant which is having so many nutrients, so many beneficial things, that it is improving our cardiovascular function, and at the same time it is lowering down our blood pressure, anti-diabetic, anti-hyperlipidemic, and even it is preventing cancer. Then what actually the problem is? If we are reviewing the data from FAO start from year 2001 to 2021, we will be noticing that there was the area that is harvested under garden pea for cultivation is something like 16 lakh hectares, and in 2021 it was increased to 25 lakhs hectares. And simultaneously, when we compare the area harvested with the production, we will finding an increment from year 2001 to 2021. That is, the production was 1 crore in 2001, and in year 2021 the production is increased to 2 crores. But when we look down the data is taken from globally area, the bee cultivation throughout the world. So when we are looking at the yield of green peas globally, in 2001 there was 77,000 hectogram per hectare of yield of garden pea plant, and when compared to 2001 it is 79,000. So there is a small amount of improvement is there over the past 2 decades. The same pattern that I have showed you, that globally this is happening, and in India also the same thing is happening. Here we have increased the area in 2001. When we are seeing the area we are cultivating for garden pea is 3 lakh, and it was increased to 5 lakhs in the year 2021. And when we are seeing the production in our country was something 30 lakhs, and it was increased to 58 lakhs. So when area is increasing and the production is increasing, but the net yield of garden pea in our country also in 2001 we are seeing it was 96,000, and now in year 2021 it is something 1 lakh. So there is not significant improvement. So the data clearly states that we are increasing the area and we are increasing the production, but when we are coming into the yield, there is no increment in that yield. So what actually the reason behind that one? The reason behind that one are the biotic and abiotic factors. These biotic and abiotic factors causing the decrement in the production of garden pea. So when we coming down to the biotic factors, especially biotic fungi named Iricipe piscis is known to be causing 25% to 50% of loss to the global production, to the production of garden pea. So this is how how the progression of the garden pea occurs, and these images are taken from our old life science building. Here I have grown the garden pea plant. This is a 30-day old plant, and on 40-day the symptoms of infection appear. So this infection, the appearance of the infection is easily recognizable by seeing the plant itself, and white fuzzy patina will be there on the leaf, and within a short period of time, I mean in 50 days, the complete field was destroyed. So if this pathogen is getting appropriate cue from the environment, within a short state of time, it is causing damage to the completely crop. So how this pathogen is working? The pathogen, the spores of this pathogen falls onto the leaf through wind. Upon falling into the leaf, these spores start germinating. The first thing the spores will form is the primary germ tube. After that one, it will form a dome-shaped structure called aprisoria. To this aprisoria, it will form a peg-like structure. So with utilizing this peg, it started penetrating into the leaf. So upon entering into the leaf, this pathogen will form a secondary germ tube, followed by formation of haustoria. So with that haustoria, it started sucking the nutrients from the garden pea plant. So these are the images that have taken for the garden pea biotic pathogen Neuricepae piscis utilizing same technology. So here you can see that the spores which are not in germinated stage, and here these are the spores which have formed the mycelia. The thread-like structure what you are seeing are the mycelia. And here in this image, see, these are the spores which are penetrating inside the leaf. So background is the leaf.
So this is your own data?
Which one?
This one?
Yes, sir.
Okay, because what is happening is you are mixing the online data versus this, so we should know where your data.
Yeah, this is my own data.
Yeah,
This is my own data. So then when we are so what are the different strategies that we are using to safeguard the plant against this pathogen? The first is that one, genetic resistance. We are having some plants, garden pea, some cultivars in garden pea which provide monogenic resistance. This monogenic resistance is provided by 3 different type of genes. One is ER1, ER2. These 2 are recessive genes, and one gene is called ER3 genes. That is a dominant gene. So these 2 genes that are providing resistance, they provide resistance based on particular location, and that also it is providing resistance to particular strain. So it is not providing complete protection against the pathogen. And apart from that one, when we come down next to what are the different strategies that we are using to safeguard the crop, the next thing is we are using fungicide. Fungicide like the combination of sulfur and dinocop we are using to protect against this biotic pathogen. But as you are well aware that the fungicide, when they are used in excessive quantity, it is having an adverse effect on environment. It is causing damage to flora and fauna. And the next method or way in which we can safeguard the garden pea leaf, we can increase the garden pea production and protect the plants is the use of incomplete genetic resistance. Incomplete genetic resistance in which we have to incorporate the minor resistance genes into the crop. But identifying minor resistance genes itself is a difficult task. So all these things have been stopping the production of garden pea plant. So the microRNA is a small 20 to 24 nucleotide sequence that is non-coding, and it plays a significant role in post-transcription gene modification. So gene regulation. It complements the mRNA, and it based on the complementarity with the mRNA, it either causes repression or it causes translation inhibition. So this plant mRNA involves in various biological process and in signaling development stress response. So after microRNA, there is microRNA like RNAs. I am just introducing this. MicroRNA like RNA is found in biotrophic pathogen, not in garden pea plant. So these are similar to canonical microRNA, and the sequences is 18 to some 20 nucleotide. So microRNA like RNA are short regulated in non-coding sequences. They play significant roles in controlling gene expression, and the biogenesis of mRNA relied on the cleavage of dicer enzyme and the activity of BCL-like enzyme. It plays a significant role in fungal stress response and various physiological processes. So,
did you mention that microRNAs are absent in post-plant within the garden tree?
no, there are two different things. As I said that my area of interest is host and pathogen interaction. It first in host microRNA present, but when we are seeing the pathogen, microRNA like RNA is found. So I found it in pathogen, fungal pathogen. So only few people have did that work on they have identified microRNA and like RNA. So far, no study have been
How
done.
similar those two are? When you find pathogen to the plant, are they similar?
The nucleotide sequences was different between this one and that. So actually, this when we are having microRNA, which is having some 18 to 24, but this sequences is having a really less sequence length, and it is the part of the canonical microRNA only. So researchers have extensively worked on the transcriptome of garden pea plant, and they have worked on various, pathogen, fungal pathogen, and in various plants. Complete transcriptome data is available to us, but all the studies conducted on powdery mildew host and pathogen lack comparability, as the studies are focused on various hosts and on various powdery mildew pathogen at different points. So to enhance the production of legume plant throughout the world, there is an essential need to inspect and identify genes that we can use in genetic engineering. So already Campo et al. Has reported that OSMI-MR overexpressed improve the resistance of plant in Magnaporta, and Sharma et al. Also has reported that OSMI regulate heat, salt and drop stress. Similarly, Su et al. Reported that one MI-520 is involved in regulation of stress-related genes in cowpea and can serve as a biomarker. And similar to that one, Wang et al. Has reported in Puccini's. So now I am coming to the mRNA part. So to what extent they have made a discovery? In my mRNA part, Puccini's study when reported by Wang et al. That during the wheat and wheat PST interaction, the study reveals that PST MR1 is a key pathogenic factor in Puccini's stelliformis that work as an effector molecule. So they said that the mRNA is work as an effector molecules when these effector molecules will suppress the wheat defense response. Similarly, they have reported Zhu et al. Reported that PMMIR16 adaptive regulate the virulence genes expression. So mRNA in this particular in apple valsa canker, the disease caused by valsa mali, the MILRN, MIL-R6 adaptively regulates the virulence gene expression. So this biotic and abiotic structure stress, this biotic and abiotic stress has been regulating the production of garden pea. To minimize the damage caused by biotic and abiotic factor, there is a need that we should study the